Hello, my name is Vicki Horn and I'm the president of Chicago Cays Community. On behalf of our community, I would like to welcome you to today's event. Our Chicago Cays represent over 3,000 out of 120,000 plus alumni throughout our domestic Cays communities. Although we're not able to meet in person, we look forward to getting to know you through our virtual programs this year. If you are in the Chicago area and would like to get involved, please get connected to us. We will be sending out a thank you email after the event with a link to our home page, which will include our contact information. We are thrilled to have alumni, families, and friends of the U from all over the country joining us tonight for this great collaboration with alumni and faculty from the Rosenstiel School of Marine and Atmospheric Science and the Shedd Aquarium. I'm very excited to hear from our fantastic panel tonight, and I hope you are too. Now, before I turn things over to our speakers, I would like to get a few logistics with you to ensure everything goes well and we get the most out of our time together this evening. The chat feature will be available, so feel free to introduce yourself, add comments, and chat with us throughout the program. Should you have any questions, please submit those via the question and answer feature located on the bottom of your screen. And we will do our best throughout the event to answer as many questions as possible. Now, let's get the program started. I am eager and pleased to introduce our moderator this evening, Ms. Cheryl Mell. Cheryl is the Senior Vice President of Conservation Partnerships and Programs at the Shedd Aquarium, where she oversees the Shedd's diverse conservation efforts, connecting audiences to outreach and initiatives including field research, direct action, advocacy, policy, and conservation travel experiences. Cheryl has 33 years of experience in museums, science, and education. She received both her Bachelor of Science degree in Biology and Marine Science and a Master's in Education, Biology and Secondary Education from the University of Miami. Cheryl, we are so lucky to have you to lead this important conversation tonight. Thank you, and please take it away.
Sure. Thank you, Vicki, for that warm welcome. I appreciate it. And thank you all who are all on this call attending. This is a full circle moment for me. As an undergrad in marine science at the University of Miami, I was able to go on many research cruises aboard Shedd Aquarium's research vessel to help my professors study aquatic life, specifically in the Bahamas. Tonight, you'll hear more about our collective work that is being undertaken. So let's get the night started. Our topic tonight is climate change and the importance of coral reef conservation for our planet. We will discuss the importance of coral reefs, the impacts of climate change on these ecosystems, and new conservation research, some of which is happening aboard the Shedd's research vessel, the Coral Reef 2. I'd like to introduce to you now our three panelists. Our first is Dr. Andrew Baker. He is a professor at the University of Miami's Rosenstiel School of Marine and Atmospheric Science. His research investigates the impacts of climate change on coral reef ecosystems, focusing on the adaptive response of corals to warming temperatures. His lab is currently developing and testing ways of manipulating corals to make them more resilient to heat-induced episodes of coral bleaching, and how to improve coral survivalship through assisted migration and/or selective breeding. Dr. Baker received his undergraduate degree from Cambridge University, graduating with a double first in natural sciences and zoology, and received his PhD in marine biology from the University of Miami's Rosenstiel School of Marine and Atmospheric Science. Next, I'd like to welcome Dr. Ross Cunning. Ross is a coral research biologist at SHED's Daniel P. Harther Center for Conservation and Research. He studies coral reefs in the Bahamas to understand how to sustain reef ecosystems in the face of changing climate. Dr. Ross Cunning's cutting-edge research has made him an expert in the relationship between corals and their symbiotic algae, which provide corals with food and give them color. Dr. Ross earned his PhD in marine biology and ecology from the University of Miami's Rosenstiel School of Marine and Atmospheric Science. He has a bachelor's degree in biology and environmental science from Duke University. And forgive me, Ross, forgive me, Dr. Ross Cunning, I call you Ross at work, so I was a little casual there. Our third panelist is Dr. John Parkinson. He is an assistant professor of integrative biology at the University of South Florida in Tampa. His research involves diving on reefs in the field, maintaining coral and algal cultures to experiment within the laboratory, and translating his findings into guidance for coral restoration and conservation practitioners in Florida and beyond. Dr. Parkinson received his Bachelor's of Science in Marine and Atmospheric Science from the Rosenstiel School of Marine and Atmospheric Science and his PhD from Penn State University. All right, I want to thank my panelists, all of them, for joining me tonight. And I'm going to start with you, Ross. Getting us started here, we've seen a lot about corals in the news. How are our coral reefs doing?
Thank you for those introductions, Cheryl, and I'm excited to kick things off. And you're right, we've seen a lot about coral reefs in the news recently, but unfortunately that's because they're in trouble. So our reefs are threatened by a number of different local and global stressors. The most important of those is climate change. And the way climate change impacts reefs is when the waters that reefs live in get too warm, a normally healthy reef that looks like this one here on your screen, full of dark brown pigmented corals, will actually turn white. And this is a response to stress known as coral bleaching. So this is what a bleached coral reef looks like. And if that bleaching event is too severe or the water temperature remains high for too long, then those bleached corals will actually die. And what you end up with is a dead coral reef like this one, where you have these skeletons of corals that were once living but are now eroding away and getting covered in algae. So this coral bleaching is a really big problem for reefs. And what bleaching is it's a disruption of what's a delicate symbiosis between corals and these microalgae. So a healthy coral actually has millions of these single-celled algae living inside of its tissues. And they play a really important role because those algae, like other plants, are harvesting energy from the sun and then providing that in the form of food to the coral itself. And when the water gets too warm, this delicate symbiotic relationship actually breaks down and those microalgae are expelled from the coral tissue. And what's left behind is this white coral skeleton that you can see through the bleached coral tissue. And this coral, without those algae providing it food, are actually now in a state of starvation. And that's why if this bleaching lasts for too long, that those corals will die. And unfortunately, we've seen recent rapid warming in our oceans that's leading to more frequent and more severe coral bleaching events. And you can see in recent years, when we have these anomalously high sea surface temperatures, we have global coral bleaching events where reefs around the world will suffer large extents of bleaching and mortality. And unfortunately, with these temperatures predicted to continue rising, we're expecting even more frequent and severe bleaching episodes. So more of our reefs around the world are going to be transitioning from this healthy state on the left with dark brown living corals to this bleached state and then dead coral reefs. And unfortunately, once those corals die, the whole ecosystem is threatened because those skeletons and that structure that they build start to break down, and we lose the habitat that all the other animals and organisms that live on coral reefs depend on.
Ross, this is it's breaking my heart a little bit. Obviously, this is not so good. So for those of us not in Miami or not living close to reefs, can you tell us how that impacts us and how Shed Aquarium and others are contributing to coral conservation?
Absolutely. So these reefs are actually really, really important to all of us. So they provide a number of really important ecosystem services. First of all, they're one of the most biodiverse ecosystems on the planet. So there's millions of different organisms of life that call reefs home. And a lot of those are really important to us. A lot of seafood that we eat comes from coral reef environments, animals that live on coral reefs or spend part of their life on coral reefs. So that affects all of us. A lot of other organisms that live on reefs are important sources of medicine. And in fact, a really timely example of that is the drug remdesivir that's been used as a treatment for COVID-19 is based on a compound that was originally isolated from a sponge from the Caribbean. So a lot of these organisms are important in ways that we don't even yet understand. For those of us that live closer to reefs, they're also the first line of defense against erosion and storm surge and hurricanes. So they provide really valuable protection of our coastlines. And they also drive really important tourism economies. So altogether, these ecosystem services of reefs generate billions of dollars of value for the US economy every year. And at SHED, even though we don't live right next to reefs, we play an important role in conserving them because we teach people every day about the importance of reefs. And we have 2 million visitors come through our doors each year, and we're able to connect with that audience and teach them about the importance of reefs and get them to have a stake in the game of protecting these ecosystems. And because they're threatened by the global problem of climate change, the solutions have to be global too. And that depends on having audiences in Chicago and all over the country and all over the world buy in to the importance of coral reefs and their conservation. But we do more than just that too, because we have this fabulous research vessel, the Coral Reef 2, whose home base is actually in Miami. And for those of you joining us from Miami, if you've driven over the 12th Avenue Bridge anytime recently, you may have seen the vessel there in the Miami River. So we have been using this research vessel to conduct coral reef research and restoration trips recently. And here's a video that I'm going to play for you that gives you a little glimpse into what one of these trips looked like last year. So here's actually John and I on the boat conducting a research trip last year where we were working on coral restoration with some of our partners in the Bahamas. And this restoration actually consists of growing coral fragments on these PVC underwater structures that we call trees. And these corals grow really rapidly on these trees. And once they're large enough, we can then fragment pieces of those and plant them out onto degraded reefs. So this is what some of the coral restoration in action actually looks like. And so we're very excited to be working with multiple partners in the Bahamas on these activities here. Some of them here, they come from academic institutions, from aquariums, from non-governmental organizations, and all these partners are critical to making this research work. We're actually very excited now that we're going to be taking this to the next level starting next year with a new grant from the National Science Foundation. So we have a $1.3 million budget in this grant to support collaborative research among the University of Miami, Shedd Aquarium, University of South Florida, as well as the Perry Institute for Marine Science, an NGO operating in the Bahamas, and the University of Southern California.
That's so great, Russ. I know you worked hard on it and the rest of the panelists did too. I'm going to switch to John. John, can you tell us about this new research grant? What are the main goals and objectives?
Absolutely. So Ross alluded to these trees underwater that we can use for conservation purposes, and we get to move corals around from location to location. And that's really also a great scientific tool. And so the goal, what we really want to do is make sure that our restoration works well, that we're moving corals that are worth moving around, the ones that are going to survive well, grow well, and hopefully in the future be tolerant to climate change. And as a part of that, as we move these corals around, what we get to do is build essentially what's a twin study underwater. If you have twins and they grow up in different environments, even though they share exactly the same DNA, you might end up with different outcomes in terms of how they perform in this context for corals. And so with corals, we can break them apart and suddenly we have two twins and we can leave one in its home location and move the other to a different one. And that's what this chart is showing that you can see right now. We have 4 nurseries throughout the Caribbean, and the idea is to basically transplant all the corals and we have twins in the audience, that's great too. We want to transplant these corals to each of those locations, so we have all corals represented at all locations. And then we get to monitor how they grow and perform over time. And that gives us a sense of which corals are the ones that are going to be most valuable for conservation efforts in the future. What that looks like, one of the things we really want to monitor is how well they grow, right? That's one of the easiest things to check. And corals can grow pretty fast. On the left here, we've got a nursery that we've just started, so we've added fragments to it that are relatively small. And then in under a year, you can get a nursery like we have on the right that Ross is looking at, a nice big bushy pile of corals that needs to be outplanted onto reefs to help them survive. But over the course of these months, we can measure them and compare growth rates amongst different colonies. And that's what we've done. So this is the location of our 4 nurseries in the Bahamas. And the map here, in addition to showing where those nurseries are located, also provides you a glimpse of the different temperatures, the average temperatures in the Bahamas in the ocean. And so we're sourcing colonies, coral colonies from places with different temperature regimes and looking at how they do when we move them around. And so far, we've seen some pretty interesting results. And so that's what the graph on the right is showing. Basically, it's 4 different coral colonies, and you've got data from each of the different nurseries where those colonies have been growing. And if they're at their home nursery, their growth rate is depicted as a blue icon. And if they're away at a different nursery, their growth rate's depicted in red. And so on the top 2 corals, you see they have a relatively low growth rate at home, but they did well when we moved them to cooler environments. And the 2 corals on the bottom, well, they were really well adapted to where they were growing initially, their home location. And so this is really telling us that both the corals themselves, their DNA, their genetic makeup, and the environments that we transplant them to, both factor into how well those corals perform, which means that moving them around is actually potentially a really good way of creating reefs in the future that will be resilient to climate change. Now, there are other things that we want to measure, not just the growth rate. Heat tolerance is obviously a really important one when we're thinking about temperatures increasing in the water. And so towards that end, we've built these really awesome mini aquaria that we can take out onto the boat, and we can clip fragments from our corals in the nursery, bring them onto the boat and stick them in the aquarium and maintain the temperature in those tanks really precisely and manipulate them over time. So we can ramp them up by a degree or 2 or 3 over the course of several hours. Now, in the process of doing this, we're measuring the coral's performance by looking at how well their algal symbionts are photosynthesizing. And once we do those measurements, we can plot them on a graph. And again, that's what we've got up here on the left. So these are different groups. You can imagine there are 4 different coral colonies, and some that are very tolerant down to some that are not. And what's depicted is really the point at which those corals photosynthesize at half the capacity that they could under normal circumstances. And temperature increases on the right. And so in the top left, we have some corals in that red group that are actually really thermally tolerant. They can handle up to 38.7 degrees Celsius, which is very, very hot. And even at that point, they're still above 50% capacity in their photosynthesis, which is, again, very impressive. And so we'll be doing this across the Bahamas, not just at the nurseries, but at all of those different places we've indicated on the map to get a really broad sense across the entire region of how much variation there is in thermal tolerance. And then we'll pick which colonies are ideal for different locations from that.
Don, this is like really amazing. Thank you for sharing. But I have to ask, like, can we really expect to save coral reefs this way, like by outplanting these best corals?
That is the hope, and there's early indications that it will work out. And it's not just outplanting coral fragments. There are additional steps that we're taking and that this process will inform. For example, improving coral reproduction in the future. So the corals that we study actually reproduce only once a year. But what we can do is when they release their sperm and their eggs into the water column is we can actually collect them. And by monitoring which corals are performing well, we can actually crossbreed specific colonies and do a sort of selective breeding like we do for, say, cattle to get bigger and fatter cattle, right? We can selectively breed for things like thermal tolerance if we know which corals to cross. And ultimately, once the corals are reproducing in the future, that will create more diversity, more colonies that are heat resistant. And the idea is we'll be able to keep corals going even as climate change is causing population declines. In the future, things will get better. So in addition to our monitoring reproduction, each of the colonies that we're going to be sampling from will also be taking samples of their DNA, and we'll be sequencing the genome for every individual colony, all the ones that we've tagged and taken heat measurements on. And what you can do with that information, once you have genomes across all of these different organisms, is we can compare them to each other and look for genes that are associated with heat tolerance. And here's an example from a study that was done very recently in the Great Barrier Reef, where you have a very similar situation to what we have in the Bahamas. There's this heat gradient that you can see on the map. There are a bunch of different reefs there, and they had different levels of bleaching at those different reefs. So they sample colonies, they sequence the genomes, and then they're able to look at the individual chromosomes and try to find genes that were found mostly in corals that tolerated that heat stress well. And here's an example of just two that are associated with molecular processes that make corals more resilient. And the idea is that in the future, if we know what those genes are, we might be able to manipulate them. We could, for example, use CRISPR or other methods that we have in molecular biology to make those genes more abundant in the populations that we're trying to breed for future conditions.
This is super interesting. So the next question, I'm going to go to Andrew. Does knowing what genes control heat tolerance mean that we can edit genes in corals? Are we like really at this point where interventions like this are necessary?
That's a great question. Well, hello everybody. I'm joining you from sunny Miami. Yeah, Cheryl, we've really hit the point, I think, where interventions like this are starting to be considered. And in fact, just last year, the US National Academy of Sciences convened a sort of a special committee to review all of these different ideas and options and try to figure out which ones are best suited for applying or testing. And I was a member of that committee, and so we had this opportunity to review all of these things. And we really sort of grouped things into 4 different categories of interventions. We've got sort of population and community level interventions, which is this idea that we've just heard about of moving corals around and exploiting the fact that corals are naturally adapted to living in warmer conditions in some areas, and perhaps we can move those corals or introduce them into areas that are slightly cooler but are expected to warm, and that's one idea. And there's other kinds of interventions like physiological interventions. This is sometimes expressed by the idea of stress hardening corals. If we can sort of grow corals and kind of toughen them up in slightly more stressful conditions than they're used to, that will actually help them survive when they get a bleaching event or some other stressful condition. And as John just mentioned, there are other interventions like genetic and reproductive interventions. This includes things like selective breeding. It includes cryogenics, freezing coral eggs and sperm, and using those to basically generate the next generation of coral recruits that we've selected the parents from, and use those coral babies to sort of reseed reefs and introduce more genetic diversity. And then even in some places like Australia, considering environmental interventions, where one example is marine cloud brightening, where we're actually artificially seeding cloud formation offshore of the Great Barrier Reef, with the idea that increased clouds will actually decrease the heat load on the water that is about to hit the Great Barrier Reef, and that will sort of alleviate the environmental conditions that trigger bleaching to begin with. So obviously there's many different approaches, and some of them we're more ready than others. But there's a lot of different ideas out there, and it's really an exciting time actually to be a coral biologist, because we have the opportunity to apply sort of 21st century science to this 21st century conservation problem.
Yeah, honestly, I'm thrilled to hear about all of these research that is going on for corals. Can you tell us, like, what are other coral conservation projects going on right now at the University of Miami?
Yeah, so what the University of Miami has, obviously, is that all of these interventions, what they have in common is that you really need state-of-the-science laboratories and facilities in the vicinity of coral reefs so that you can rapidly test and deploy these interventions and sort of monitor their results. And that's exactly what we've been doing. So in addition to the projects that you've heard about, we're also considering many of these other ideas, including introducing corals from the Bahamas into Florida to replenish our own genetic stock, and also vice versa, to help boost the resilience of corals in the Bahamas by introducing Floridian stock. But doing this in a controlled scientific way where we can sort of monitor impacts and sort of judge the cost benefits. In my own lab, we're also doing other investigations such as ex situ coral spawning. So this is the artificial spawning of corals in the laboratory that will help us with these selective breeding efforts. But really the message here is that there's probably not going to be one single approach, one sort of silver bullet that's going to solve these problems. Each reef is going to probably need an intervention that's perhaps slightly different from a reef in another area, so it's very context specific. And what we really need to be doing right now is almost taking a page out of the engineering book and realizing that we need to come up with solutions, and we need to come up with them really fast. And that means identifying the things that work as quickly as possible, as well as the things that don't, this sort of fast fail approach. And I should also mention that all of this really just helps us buy time for reefs. We still have to confront the larger problem head on, which is that of climate change driven by greenhouse gas emissions. And unless we can get those underlying threats under control, all this does is buy time for reefs, which they need right now, but it's not a solution in and of itself. Really what we have at the University of Miami is this opportunity to really build on our history. Some of you who are joining us might know that the University of Miami's Rosenstiel School was actually founded by a coral reef biologist back in the 1940s, in 1943, Dr. F.G. Walton Smith, and he actually wrote the book on Atlantic reef coral identification just a few years later. And here we are, over 75 years later, and our motivations for studying coral reefs, are still grounded in that scientific curiosity, but they're really fueled by this urgent need to try to confront the coral reef crisis. And obviously our location at the northern end of the sort of largest barrier reef ecosystem in the continental US just makes us particularly well suited to be able to capitalize on this opportunity with partners such as SHED, who are operating vessels in waters nearby. And it's really an international effort amongst scientists and among institutions, because as I've sort of alluded to before, no one solution is likely to be the overall winner, and no one institution is going to be able to do it all. So it's truly a collaboration. And Miami as well, just from our own sort of selfish perspective, we have a lot to gain from saving reefs. Miami, as everyone knows, is a very low-lying city with a lot of infrastructure that's vulnerable to the impacts of storm surge and sea level rise. And in fact, having healthy coral reefs off our coastline is tremendously valuable just in terms of avoided protection from flooding. One study published just last year estimated that even in a non-hurricane year, the value of having healthy reefs off of Miami-Dade County is something like $657 million every year in avoided flooding benefits. So obviously Miami is in the process of doing a lot of planning and thinking about what sorts of engineering and cement-based infrastructural improvements can help us deal with these threats. But there's really an important conversation going on as to the role of nature-based solutions in promoting or helping these gray infrastructure, these cement-based infrastructural approaches be more effective, be more self-repairing, self-growing, and also very cost-effective. And as a result of that, we also have another project going on at Erasmus right now that exploits the fact that we have one of the world's only wind wave tanks. It's called Sustain, and it's a facility that's able to generate hurricane category 5 force winds as well as waves. And we've been using this facility, which is over at the Rosenstiel campus, to actually measure the ability of coral reefs to mitigate wave action, to reduce storm surge, and then apply that data and information to modeling efforts and implementation that's actually happening off the city of Miami Beach right at the moment. So it's very much a a practical solution that's really near the hearts and the pocketbooks of many folks living in Miami today.
Thank you, Andrew. That was wonderful, and I'm going to take you up to come see that wave tank in Miami next time I'm there.
Please do.
Yes, so I'd like to actually thank all of our panelists for your presentations and ask you to join me on screen right now. We did get some questions in advance, and I know folks have been submitting them during this program. So I'm going to just kind of call on each of you, group some that are like to each other. And Ross, I'm going to put you on the hot seat first. So Ross, two questions. How does coral fragmentation and transplantation make a difference in coral restoration? And have the reefs we have been planting shown positive signs of developing into larger reefs?
Yeah, absolutely. So coral restoration by planting out fragments has been one of the dominant method of reef restoration that's sort of been set up and been implemented thus far. But and now tens of thousands of corals are outplanted onto the Florida reef tract alone on an annual basis. So it is it has a lot of use. There's widespread effort going into restoration of this method, but you have to ask yourself, at some point, are you really going to be able to restore reefs at such a massive scale, like the Great Barrier Reef, just by planting out corals? So there is an urgent need to find new solutions to scale those methods up. But in localized areas where this restoration does take place, it's been very successful. So places like Belize, where restoration has been going on for a long time, you now have reefs comprised of large thickets of these branching staghorn corals that were all outplanted onto those reefs. Like John mentioned, those are fast growing corals, and when you get enough of them put out to a location over enough time, they can build back a coral reef. So there are examples of places with enough effort put into that kind of restoration that it's very successful. Another important milestone, besides just wanting those corals to grow and form large reefs, we want them to reproduce, right? We need those corals that are restored onto reefs to start reproducing and self-seeding and sustaining themselves. And just this year in Florida, actually just a couple of weeks ago, was the annual coral spawning event, and researchers observed a lot of corals that had been restored onto our reefs now that they are spawning themselves and contributing to that reproduction that then will sustain the ecosystem. So there are definitely positive signs and success stories that have come out of, from that kind of restoration.
Okay, good, thank you. John, I'm gonna hit you up next. So what organizations and technologies are proving the most effective in restoring coral systems at scale? And what are you doing to support captive breeding programs and captive spawning of corals?
Sure. Well, I have a sort of interesting history because prior to where I am now at the University of South Florida, I was working with an organization called SECOR International, which does a lot of great work on specifically targeting sexual reproduction of corals, and that's so getting spawning to work, and then after the corals spawn, trying to move those larvae back onto the reef in targeted locations where they're most needed. And so SECOR has made a lot of progress on that front, and they're, it's a huge network of people who are working together. So there are collaborators in the Great Barrier Reef that right now are doing similar things, but they have these really interesting slicks of coral spawn when they're released. They form these large slicks on the ocean surface. And they've actually been using robots recently to move through those slicks and collect gametes and then redistribute them onto the reef. So there's a lot of motion in the direction of sexual reproduction because it is scalable, more scalable probably than outplanting individual colonies. The trade-off is a lot of those individuals end up expiring. That's just the nature of things. Corals release a lot of gametes and sperm and eggs to sort of compensate for the fact that many of them will disappear. But this is a challenge that we're meeting head on and making definite progress in. And then what I'm doing now to help, I guess, is this grant and this work that we've been doing in the Caribbean. It's been really great to get onto Shed's boat and to go out into the field and be able to set up some of these nurseries and broaden some of the nurseries from what they were originally to what they are now. Just moving those corals around is in and of itself a conservation effort more than just a scientific effort, which has been great. And then we work closely also with the Florida Aquarium, and they've been pioneering in ex situ, so in aquarium spawning, along with the Hornman Museum, I believe in the UK, to really target a lot of different species and get them all to reproduce in the laboratory setting. Prior to their efforts, we mostly have to go out into the field, but now it seems like we are going to be able to have at least many more species than previously grow and reproduce within an aquarium, which is fantastic.
I love hearing this, and I know we have a coral research cruise the first week of October on the research vessels, so excited
Awesome.
to have you all. Ross, I'm going to throw one more back to you right now, and then we'll go to Andrew. So Ross, with increased levels of bleaching, how has the progress with the clades of zooxanthellae led to more resilient corals?
Right, so the question Cheryl's asking about these zooxanthellae, so that's another word for those microalgae that I mentioned that live in this symbiosis with corals. And it's actually not just one type of microalgae that can form these symbioses with corals. There are many, many different types, different species of these microalgae, some of which have very different characteristics from one another. And depending on what species of these microalgae that are found inside of a given coral, that actually is a big determinant of how sensitive that coral might be to bleaching. And there are some corals that can actually bleach through which they lose those microalgae, but then they can actually recover and become healthy again. And they recover with a different species of microalgae that's actually more tolerant of high temperatures. And so by going through that process of changing or shuffling the species of microalgae that they associate with, they can actually become more heat tolerant. So that's been an active area of research actually in Andrew's lab, and I worked on that during grad school there. John's worked on that as well. It's been a highly active area of research because it is kind of one of these glimmers of hope. It's a mechanism by which some corals may become more resilient and be able to survive in scenarios of ocean warming. It's not something that all corals can do and that's going to be able to save corals, but there are certain corals that do exhibit this phenomenon that we're really interested in, and we hope that and other mechanisms that these corals have to sort of adapt and acclimatize to changes in their environment will come into play and help them get through some of the stressors that they're facing.
Thank you. Yes, I appreciate that. Okay, Andrew, you're in Miami, a little jealous right now. So can you tell us, like, someone asked actually, where are the coral reefs in the Miami area? Are they in danger? What are plans on a federal, state, and county level to reverse the course of damage that's already been done?
Yeah, so there are coral reefs in Miami, and in fact they're fairly extensive, and there's some evidence that in fact corals are slowly beginning to move northwards as the climate begins to warm, or at least some species. They're not as extensive as they are in the Florida Keys, but as I mentioned before, they're still tremendously valuable, not just in terms of coastline protection, but also in terms of biodiversity and tourism and recreational fishing opportunities. Reefs in Miami, just as the rest of Florida, are under threat, and in fact a new threat that has arisen in Florida and has now actually begun to spread out of Florida is a novel disease called stony coral tissue loss disease, which actually appeared off Miami as early as 2014 and has since spread through the Keys and into the sort of nearby nation states of the Caribbean. And this disease has actually had devastating impacts on corals of a variety of different species throughout the area. And so in response to that, and this is a threat in addition to that of climate change and all the other local stressors, it's really triggered quite a significant effort from the state and from the Florida Department of Environmental Protection to try to fund some of the interventions that we've been talking about, looking at the pathology of the disease, the epidemiology of the disease, how it's spreading, how it's contracted, what the sort of what makes a coral susceptible and what makes it resistant, and then restoration efforts to try to identify populations of corals, genotypes of corals, types of species of corals that are resistant that can be then either restored using the sort of asexual approach, but also potentially interbred against other populations that are vulnerable to try to inject some of those potentially resistant genes into the population. So there's lots of ideas out there. And as I mentioned before, it's a very active area of research. It's kind of an exciting time, despite the fact that, unfortunately, this is a response to a crisis. But there's a lot going on.
Absolutely. It does sound like there is a lot going on. John, I'm going to put a few of these together and ask you, so we have some divers, what can an average South Florida scuba diver do to help this cause? How can everyday people get involved in the effort? And what is the most important action we can take as individuals, I think living in Florida or some of us here in Chicago, to do to protect reefs?
That's a great question, and reefs are out there. They're in the water. They're hard to see, and I think sometimes that's why people don't think about them all that often. But one of the most important things you can do if you're interested in this topic is to talk to people about it, particularly if you're not by the coast and you can't take someone out on a boat to go snorkeling. Just the more people are aware, the more people go to fantastic places like the Shed Aquarium that give you a glimpse into what's in the ocean and can inspire people. Even, Finding Nemo is a big positive for coral research and awareness about corals. But there are some more specific things if you're a diver that you can do. For one thing, if you're in the ocean and you see a coral, don't touch it. I try not to, right? Don't stand on corals. These are sort of simple, basic items. But these nurseries, there's a whole network of them throughout Florida. And if you're interested in them and you want to help them out, there are ways to do that. The Coral Restoration Foundation is in Key Largo. The University of Miami has its Rescue a Reef program. So their nursery, volunteer divers go in with them all the time, get a chance to check out the structures, clean them off, and learn more about the process there. But those are, there are some other things, don't eat fish that are not sustainable, and the general overall issue of greenhouse gas emissions, which I know is a structural problem more so than a personal problem, but every bit that you can do to help is appreciated and should help corals in the long run. But I think most importantly, it's inspire other people, get people to know about corals and care about them, and then we can hopefully make some of the systematic changes that need to happen.
Absolutely. Thank you. Okay, we got a few more here. Andrew, I know you talked a little bit about our partnership and other aquariums, Florida Aquarium, who are working around the country with University of Miami. How is everyone helping each other out? Is everyone working collectively? It sounds that way, but I think folks want to hear a little bit more.
Yeah, so obviously, in addition to Shed and the Florida Aquarium, which John mentioned, we're also have been working quite closely with the Frost Science Museum in Miami. We've had a partnership with them for several years. It's sort of leveraging the fact that they have this public audience. And in fact, for a while, we had a laboratory there where we were actually testing some of these interventions, and members of the public could actually see those interventions in action being tested as we were gathering data. I noticed in the Q&A that people were wondering about collaborations with Australia, and that's certainly happening. Australia has had a sort of its own version of the US National Academy of Sciences Committee, where they've also convened their experts, and we've had some of those Australians part of our panel, and vice versa. So there's a lot of exchange of information. In many ways, the Australians, have this problem on a scale that's an order of magnitude 10 times bigger than the problem we have in Florida, just because of the size of the Great Barrier Reef and its relative isolation. in some ways, you know, and that's really gets back to what I said about the location of Miami. It's easy for us to be able to test and develop these interventions on our own, in our own backyards, because we don't have to get on a research vessel and cruise for hundreds of miles to get out to the reef, the Great Barrier Reef. Yeah, there is a lot of information exchange, and there's a lot of really a kind of a sense of community, and the idea that coral restoration in all of its forms, not just the planting out of corals, but the ultimate goal is how do we get self-sustaining, reproductively mature populations of corals that are able to, you know, use the power of nature to replenish these reefs through their own methods of natural reproduction. And I'm really happy to say that just this last month, my colleague at Rasmus at the Rosenstiel School, Diego Lehrman, his populations of asexually produced corals, which he's been planting out on trees for years, actually sexually reproduced for the first time just last month. And that's a really encouraging sign that ultimately, if we can help nature help itself, then we will have a fighting chance of helping coral reefs persist. But as I mentioned, it's a collaborative and international effort.
Thank you, Andrew. I'm actually going to probably stop us at this point. I do have a favor to ask of my panelists. First, I want to thank you, Andrew, Ross, and John, for a very informative conversation tonight. I know there was some additional questions that were in the queue. Oh, they just went away. Otherwise, I was going to ask you to type some answers to them. But I really appreciate this conversation, and I thank this audience so much for joining us, participating in the chat and the Q&A. I am going to turn it over now to our floor back to Vicki Horn, our Chicago Canes Community President, and ask her to give us some closing remarks. I did see the questions are back, so maybe some of the panelists can answer some of those as Vicki goes through those last few slides. And again, thank you for letting me participate. I so appreciate it. All right, Vicki, it's all yours.
Thank you.
Thank you, Cheryl, Andrew, Ross, and John. What a fantastic presentation. And thank you all for joining us for such an important, engaging conversation tonight.